Hello and welcome to my talk. I'm Sebastian Niehus from the Junior Research Group Genome Informatics at the Berlin Institute of Health, and I'm going to talk to you about how PopDel detects large deletions jointly in tens of thousands of genomes. Let us start with a quick recap of structural variants, or SVs, and why they are an important research topic. Structural variants are often defined as affecting at least 50 base pairs of genomic sequence. Recent studies suggest the presence of tens of thousands of SVs per human genome. Compared to short variants, this does not seem much, but due to their size, they affect many more bases and are therefore much more likely to be relevant for disease. Associations to conditions like Alzheimer's disease, different muscular dystrophies, or autism have been found in different studies. They are also more diverse than short variants. Sequence can be duplicated, inverted, deleted, translocated, or non-reference sequence can be inserted. Further, complex variants can be described as a combination of these relatively simple SVs. In the following, we will focus on deletions. Their size and complexity make the detection and genotyping of SVs more demanding than the well-established small variant calling, and therefore new approaches are required. One possible answer to this problem is the joint calling approach. It is defined as the integration of data from different samples in a joint detection process. This approach is already well established for small variant calling, like performed by the GATK haplotype caller. But to our knowledge, PopDel is the first tool which is capable of detecting SVs jointly in tens of thousands of genomes. So, what exactly are the differences between joint and single sample calling approaches? For the single sample approaches, the calling is performed per sample. Then, the individual call sets are merged to a combined call set, which is then used for the sample-wise regenotyping. The resulting VCF files then have to be pasted into a combined call set of the final variants. This has several drawbacks. As you can see, this type of approach requires complex workflows, and there are often differences in the representation of the SVs. The caller might disagree on the exact position or size of the variants, requiring the merging to be heuristic. Further, there is no utilization of data from multiple samples, which would facilitate the detection of shared variants. Joint calling approaches, on the other hand, look at the data of all samples simultaneously and use them in a combined detection approach, thereby circumventing the need for merging and creating a much nicer and shorter workflow. Of course, there are a few challenges for the joint calling approaches, some of which are the management of so much data at once, the scalability to thousands or 10,000s of samples, and the question of the treatment of variants with a low allele frequency. As you maybe can imagine, the low allele frequency variants require some special treatment, so they are not lost among the many samples which do not carry them. This brings us to our tool PopDel, which implements the joint calling approach for SVs. Like other tools, PopDel detects deletions by observing the increased distance between the mapped read pairs, which is caused by this deletion. As you can see in this picture, the distance between the read pair at the top of the figure increases when mapped to the reference DNA. This is the main signal PopDel is looking at. Unfortunately, the read pair distances are not some fixed value, but rather follow some distribution. Therefore, we decided to measure the deviation delta from the empirical distribution mean. Let's have a look at the following example. Here we have, in blue, the distribution of read pair distances from read pairs which are not affected by any deletion, and in red, the distribution of read pair distances affected by a deletion of size delta. The difference in the means of the two distributions corresponds to the size of the deletion. One special point of PopDel is that we create the blue curve for each sample or each read group of each sample individually by sampling from some well-behaved regions. Because we estimate the background distribution per read group, We can even process data from different sequencing libraries with different read pair distance distributions together. So let me show you how we use these histograms for calculating the likelihoods for the possible different genotypes for a set of reads. First, we have the likelihood of observing the set of reads capital R under the assumption that the real underlying genotype is G0, meaning the absence of the deletion in both of the alleles. As we assume the reads to be independent, we can calculate this likelihood simply as the product over the histogram values for the read pair distances we observed. For the likelihood of observing the given data, assuming that both alleles of our sample are affected by the deletion, assume that all of our observed read pair distances originate from the red histogram. By subtracting the estimated deletion size delta from the observed read pair distance, we can shift the red histogram to match the blue one and use this again for the calculation of the likelihood. The heterozygous case is kind of a hybrid case between those two. We expect roughly half of our read pairs to originate from the blue histogram, so from the unaffected allele, and half of our read pairs to originate from the red histogram, so the allele affected by the deletion. Consequently, we get the density values for our read pairs from the blue and from the red histogram, so from the affected and unaffected histogram, weight both of them with 1/2, and then use this in the product over all observed read pair distances. We then use the calculated likelihoods in a likelihood ratio test to assess whether a deletion is present or not, and came up with the following likelihood ratios. For the sake of simplicity, let's assume we only have a single sample. Here, we calculate the likelihood ratio lambda by dividing the likelihood of the deletion model by the likelihood of the reference model. The likelihood of the deletion model in this case is calculated by multiplying a prior for the observation of a deletion with the sum of all calculated likelihoods. Similarly, the reference model is calculated by multiplying 1 minus the prior with the previously calculated reference likelihood. This calculation now has to be transferred to multiple samples. Let capital S be the set of all our samples. Like we did before, we create the sum over all likelihoods for all genotypes, but with the difference that each likelihood now is previously multiplied with an individual weight. These weights are necessary for ensuring that we can also detect variants which are only present in a single allele, even though all other samples do not carry this variant. Now we assume that all samples are independent, so we can create the product over all of the previously created sums and multiply this again with our prior. The likelihood of the reference model is calculated analogously, but does not rely on the presence of the weights. In a final step, we then take the logarithm of the resulting value lambda, multiply it by 2, and compare to the chi-squared distribution. We decided to test for the one-sided 99.9% quantile, as this gives us robust results. Now I can show you how these calculations come together in PopDel's workflow. It consists of two main steps. The first step is the profiling step. Here, PopDel extracts all necessary information from each sample or BAM file and stores it in a small binary profile. Profiles contain, for example, the background distribution of the read pair distances and the read pair distance information for each genomic position. By storing the profiles in binary format, we can reduce the disk space to only 1 to 2% of the original size of the BAM file, and further, the profiles contain their own index for faster processing. This brings us to the calling step. In the window-wise calling, PopDel moves a 30 base pair window across the genome. In each window, it looks at the read pair distances of all profiles and uses them to initialize the candidate deletions. For n samples, this can give us up to n candidate deletions per window. This allows us to even detect overlapping deletions from different samples. Each candidate deletion is then processed in an iterative approach, which takes the data of all profiles into consideration. Here, PopDel calculates the properties of the deletion, like allele frequency and length, estimates the weights which are necessary to boost the signal of rare variants, and calculates the likelihoods for the genotypes as previously discussed. The likelihoods are then used in our likelihood ratio test to decide whether we believe the deletion is present or not. This whole process gives us a bunch of redundant deletion calls, because one bigger deletion can of course overlap more than one genomic window. Therefore, consecutive windows belonging to the same deletion are combined, and we get a set of non-redundant deletion calls. In this step, we also consider the genotype likelihoods of each sample to assign the final genotypes. The output is then written to a VCF file which contains all detected deletions and genotypes for all samples in our cohort. Now let us have a look at some results. For our first comparison, we took the genome in a bottle Ashkenazim trio and measured precision and recall in single sample calling on the Ashkenazim son and trio-wise calling. The plot shows that PopDel achieved the highest precision together with Manta, while maintaining a higher recall than all other tools. Also, you can see that Deli and Lumpy suffer from a loss of precision when switching from single sample calling to trio-wise calling. For Manta and PopDel, this effect is basically not present or very small. We also wanted to perform tests on a bigger cohort. Therefore, we relied on the 49 trios of the Polaris Kids and Diversity cohorts. All genotypes were filtered for the Hardy-Weinberg equilibrium, and then we measured the Mendelian inheritance error. As the left plot shows, PopDel detects more deletions per trio while maintaining a lower rate of Mendelian inheritance errors. For the next plot, we wanted to know how the tools perform in the case of rare variants. Therefore, we measured the number of deletions that were transmitted from parents to the child if only one of the parents carried one allele with a deletion. Ideally, you would expect that this deletion is transmitted from the parent to the child in 50% of the cases. As the plot shows, PopDel is closest to this value while still maintaining a high number of transmissions. The last plot combines these two measures. Like in the previous transmission rate plot, the lines should be as close as possible to the horizontal line indicating the 50% transmission rate, and as close as possible to the vertical dashed line indicating 0% Mendelian inheritance error rate. So the optimum of the plot is at the intersection of the dashed lines. As you can see, PopDel comes closest to that. The running times for different real data samples looked as follows. On NA12878 with 50x coverage, PopDel takes 25 real-time minutes or 58 CPU minutes, and thereby comes second after Lumpy, which takes only 18 real minutes or 30 CPU minutes. On the 150 samples of the Polaris Diversity cohort, PopDel is faster than Lumpy because, due to its better I/O management, it has a better CPU time to real-time ratio. It only takes 56 hours for all 150 samples to be completely processed. Unfortunately, Manta did not finish within the 4 weeks of available compute time. This is also the reason why we could not display results for Manta on the previous slide. PopDel has already been applied on more than 40,000 genomes at once. By using PopDel, Decode Genetics could detect an unknown rare 2.7 kb deletion that overlaps the 3' UTR of the LDLR gene, causing very low LDL levels. This suggests a high relevance for cardiovascular disease, and a paper treating this deletion is currently in submission. This brings us to the end of my talk, so let me summarize the most important points for you. We developed PopDel, the first tool for the joint detection and the genotyping of deletions in more than 40,000 genomes at once. It is easy to use, fast, robust, and has good precision and recall. If you want to know more about PopDel, please refer to the preprint on bioRxiv. Its source code is available on GitHub, or it can be installed via Bioconda. Lastly, I want to give special thanks to some people. My supervisor, Bete Kehr, Bjarni Halldórsson and the other people at Decode Genetics, Janina Schönberger, a former student assistant who has been working on PopDel, and all members of my working group. Please note that PopDel has been implemented using CGEN and that we received funding from the German Federal Ministry of Education and Research. Thank you for your attention.